Hello everyone, this is the Theoretical Doctor and welcome back to my channel. If you want to access flashcards on this topic and other videos, do click the link below. I post weekly videos every Tuesdays and Fridays, so do subscribe for more content. Today's topic will be on disaccharides. It's a continuation of the previous video whereby I talk on monosaccharides. Alright, so a disaccharide, di meaning two, is formed by the condensation reaction between two monosaccharides. So if you don't know what monosaccharide is, do check out the previous video. Mono meaning one, so it's the building block, and di means two, so it's formed by two monosaccharides with the removal of a water molecule because the reaction is condensation, and they are linked by the glycosidic bond. So it's very important to remember this definition. Okay, so some physical properties: they are sweet, just like monosaccharide, they dissolve in water, and they can be crystallized. Now for chemical properties, all are reducing sugar. So by reducing sugar, meaning that it can actually react with Benedict's solution to form brick red precipitate. Now all of it except sucrose. And it can be hydrolyzed to form monosaccharides by heating in dilute acid or enzymes. Now this differs for monosaccharide. It cannot be hydrolyzed even more because, like I said in the previous video, monosaccharide is the building block, it's the unit, so you can't break it down even more. But disaccharide, because it's formed from two monosaccharide, you can break it down. So some important examples to remember: so if you were to combine glucose and glucose, you will get maltose. Glucose and galactose, you get lactose. Glucose and fructose, you get sucrose. These are some important ring structures you should know. It's important to actually practice it. So like in the previous video, I actually put it in a linear chain, but for this you need to actually know how to draw the ring structure. So these are some examples I'll show: alpha glucose, beta glucose, and fructose. So for glucose, it's actually a 6 carbon chain, and how you draw it, actually you just put the oxygen here first, and then you link it with the first carbon, second, third, fourth, fifth, and sixth. So remember, it's actually a hexose. Now the difference between alpha glucose and beta glucose is the placement of the hydroxyl. So for alpha glucose, the hydroxyl is placed below, and for the beta glucose, the hydroxyl is placed above. You need to distinguish this properly, okay? Now for fructose, the first carbon actually is here, so the oxygen is connected to the second carbon, and the first carbon is actually lying above. So you need to remember the drawing. So it's important, just practice it, practice the drawing, and you'll remember it. Moving on to maltose. Maltose is actually when glucose combines with glucose, you get maltose. So maltose is formed from two molecules of glucose, which is bonded by the alpha 1,4 glycosidic bond. Take note of this, okay? Alpha 1,4 glycosidic bond. So you will be using the alpha glucose, 2 alpha glucose, 2 alpha glucose. What happens is that it will actually remove water. So the hydroxyl ion from 1 alpha glucose and the hydrogen from the hydroxyl ion of another alpha glucose. So water will be removed through condensation reaction, and this is where they will be bonded here. Okay, so this part here is called alpha 1,4 glycosidic bond. It's a glycosidic bond. It's alpha because the structure over here that is being used is the alpha glucose, and 1,4 because at carbon 1 of the monosaccharide, and also at carbon 4, that's why it's alpha 1,4 glycosidic bond. Next is the sucrose. So sucrose is formed when glucose combines with fructose, okay, and they are linked by alpha 1,2 glycosidic bond. Now this is also an alpha glucose, and this is the fructose. So water, because basically to form a disaccharide, you need to do it through the condensation reaction, and for condensation reaction you need to remove water. So you see here, they will remove water and they are bonded over here. This is the glycosidic bond, and it's alpha because it's an alpha glucose 1 2. Okay, 1 because carbon 1 of one of the isomer and carbon 2 of another. So it's alpha 1 2 glycosidic bond. Now some functions of disaccharides. Maltose, they act as an intermediate in the synthesis of polysaccharides. They also act as an intermediate when starch or glycogen is broken down. Intermediate meaning being in the second phase. Okay, because after this we'll be moving to the topic of polysaccharides, and like I mentioned in the other video, poly is more than 2 and this is just 2, so it's in between. So starch and glycogen are actually some examples of polysaccharide, but don't worry, do check that out in the next video. Now sucrose acts as a medium to transport carbohydrate in phloem, so basically the food in the plant. It can lower the water potential and creates hydrostatic flow in the sieve tube and stored in plant cells as food reserve. So basically that's the plant food. And now lactose, as we all know, it's found in milk. Yep, so I hope you're not lactose intolerant, because I really do love milk actually. Alright, so if you've reached this point, thank you very much. Do subscribe and share this video to those who might find this helpful. I will see you in the next video. Bye!